Hello, welcome everybody to the Kennisclip Root Development, Cement Formation and PDL. Until now we've talked about making dentine for the crown of the tooth, but when you're looking at the root of the tooth we need a combination of dentine formation, which you can see here, but we also need cement down at the bottom of here to hold the tooth in place. So let's start with root development. How do we get this bit here? How do we make the cement to hold the tooth where it needs to be? Again, let's go back to the cartoons that I was using in the previous Kennisclips to illustrate the process of embryonic tooth development, dentinogenesis and amelogenesis. Now we're getting towards the stage of, this is the end of the bell stage, and you can see what must look familiar now, dentine, and you can see enamel. In this red box is a structure which you will now get to know very well in this Kennisclip called the hairs. I'll come back to it in the next slide. The rest of the cells should look familiar still, so you still have the dental papilla cells in the middle, the dental follicle cells on the outside. These cells are needed again in collaboration with each other to make cement and the PDL. So what is this hairs thing? Why do we need it? Let's zoom in on this cartoon here to a more high power cartoon. The hairs is the Hertwig's epithelial root sheath, named after probably the man who first described it. And it refers to this little layer of cells here at the bottom of the tooth. So going up to the top is obviously where we've been talking about dentine and enamel development. And you can see here together, when the two slides come together, the two layers of epithelial cells here come together, they lie on top of each other. This is the hairs area, so this is the extension of the external enamel epithelium, and this is the extension of the internal enamel epithelium. What else do you need to know about this? So it is still counted as part of the enamel organ along with the rest of the developing tooth, and the Hertwig's epithelial root sheath, the hairs, is formed by the continued proliferation of this internal and external enamel epithelium, these layers together. Is the production of dentine by odontoblasts. And to signal the production of dentine by odontoblasts, this hairs provides a signal into the dental papilla, turning these green cells here into these orange long odontoblasts. These are supposed to be cells which start to make the dentine here. And you can see it's been deposited along the inner side of the hairs. So here, this is the hairs, still purple, and this is along the... If we then go to the next step, the result of this dentine being deposited here is actually the disintegration of the hairs. So it's falling apart. You can see it's no longer in these double layers, and the cells are starting to have some space in between them. As a result of this, the dentine, which is here, comes into contact with the dental follicle cells. So here, on the other side now, we're talking about. This contact between dentine and the dental follicle cells provides a signal for the differentiation of dental follicle cells into two types of cells. So there's two arrows here. We're talking about differentiation of dental follicle cells into cementoblasts, these brown square cells that lay flat against the developing cement, and also into fibroblasts, these long green cells which are perpendicular orientation to the developing tooth, and these are the cells which will form the PDL. There is some research actually, because some of these things we still don't really understand super well, is that epithelial cells may also have, so the remainders of these cells may also have the ability to turn into cementoblasts and fibroblasts, but that's still a little bit unclear. So we have a point now when we have cementoblasts, which are cells which will make cement. These cells now begin to secrete protein, the protein components of cement. So remembering, they will never secrete the final cement product. It's far too hard, that would kill the cells. These cementoblasts, which line this part of the tooth on the outside, so the crown is at the top here and we're on the outside of the tooth, secrete cement or the pre-cement proteins in one direction. So it's really important you remember this. I'll come back to it later for why and which other cells do not do this. So in the case of this cartoon here, if I was this cement cell here, I would be secreting the precursors of cement this way. So this little layer of cement here is what you need to focus on. These blue kind of round bits of cells, by the way, are called the epithelial rests of Malassez, and they are basically leftover epithelial cells from here which are formed into kind of cysts, and these cysts stay around, hang around in the teeth, and can be very clinically relevant for you guys as future dentists because they can form cysts which may need to be removed. Okay, so let's look at a nicer cartoon and how this looks under the microscope again. Take it back to the real life. Again here, same idea, so I hope you can see. Here is the double layer of epithelial cells originating from the inner and outer enamel epithelium, the hairs. Here is the dental papilla, which have turned these cells into, which have differentiated into odontoblasts from signals, because of signals they received from the hairs. The odontoblasts then make dentine, which is here. The dentine then stimulates the breakdown of the Hertwig's epithelial root sheath, so you can see it's falling apart here. This contact of the dentine with cells in the dental follicle here makes them turn into two types of cells. So here they are, these guys here, cementoblasts, and cells in the PDL, these fibroblast type cells. This is what I was talking about in the last slide, the epithelial cell rests of Malassez. So these are clumps of epithelial cells which will stay hanging around for a while. On the right hand side, looking microscopically, cement is a little bit trickier to see than, for example, dentine and enamel, which I always think is very easy to find. But let's have a look at the orientation of the slide. So first of all, it's usually best if you start at the bottom and find out where the hair structure is. So this is the double layer of epithelial cells, you see? So this bit here. And if you follow up, on the left side, this is bone, so this must be the outside of the tooth, just in the same orientation that you see on the left side here. And if you follow up, these long striped cells here would be the odontoblasts, making a dentine here. And if you move towards this part of these squashed cells against these kind of small squarish cells will be the cementoblasts, and the layer that you can see happening here will be the cement precursors. So like I was saying, secretion of cement in this part of the tooth around the crown of the tooth, so we're talking about around here on the outside of the tooth in the bone socket, happens in one direction, and you can actually see this in stripes in the cement. So switch your orientation now. Now we're looking at the other side of a tooth. Here is dentine, which would be more in the middle, of course, and here is the periodontal ligament. On the right hand side you would have bone, and you can see these waves of darker staining, which is related to the waves of cement protein production by the odontoblasts. So in this first wave here, the cementoblasts secrete the proteins, and then they move backwards a bit and secrete more and secrete more and secrete more. So you will see these stripes, or you can see these stripes in primary cement formation. The name that we give to this cement here, this one directional type of cement formation, is primary cement, and there is of course then a secondary cement to complement this. So here we go. What we've been talking about is making primary cement, which is on the outside of the teeth here. So if you follow my arrow, we will be talking about cement here and here around the outside. You can see that it has been labelled here acellular cement, which means there are no cells left over in this cement. So entrapped cells, cells left over, no. Talking about primary cement. There are a lot of confusing names for cement in the literature. You will hear it called acellular cement, you will hear it called primary cement, you will hear it called all kinds of things. But these are the biological characteristics where those terms come from. So are there cells trapped in the cement? No, it's acellular cement. The direction is unipolar. The main function is the attachment of the tooth to the bone, and is mostly performed when the crown enamel is finished. It happens first in comparison to the other main type of cement, but it's quite slow, and this is as a function of the unipolar direction of cement protein secretion, so only in one direction. And it is, of course, involved with the periodontal ligament, being that it's mostly on the outside of the tooth. Wrong way, sorry. Secondary cement, however, is cellular. So it's often called cellular intrinsic fiber cement, and it does have cells trapped in it, which is why it's called cellular. The direction of the fact that the cells are trapped in it is a direct consequence of the way that these cells here secrete the cement proteins, because they secrete them in all different directions. If you can imagine being a cell secreting protein in one direction and then retreating and then one direction, so you're never trapped in the proteins that you secrete yourself. When you are secreting proteins all around you from up, down, left, right, then you build yourself into the cement, which is why this type of cement is called cellular. This type of cement is more a repair function and protecting the tooth, and happens secondarily, so after tooth eruption, and it's much faster, like I said. So if you're secreting cement proteins from more than one direction, you can build up cement faster, but you trap the cells in. Cellular cement is not normally associated with the PDL. So let's go back to the diagram here. Cellular cement, this thicker layer on the outside, on the inside, sorry, underneath the tooth, and also you can have a little bit at the bottom, but it's not as associated with the PDL like the primary. We can see the difference between these two types of cement, if you look under the microscope. So again, dentine on the left, PDL on the right, acellular external fiber cement. So the other extrinsic fiber cement, so the other parts of the name will become more obvious when you get more into the formation of cement, the process. This is the picture you saw before, so these are the striations of primary cement formation. And I hope you can see in contrast here to how this cement looks. So here is dentine again. This area all the way from here to the outside here is cement. So it's a lot thicker. The secondary cement is a lot thicker than the primary cement. And what you can see here, these are the cementoblasts, all these little dark cell bodies in the middle with all the protrusions. These are the cementoblasts which have been built into the cement because the secretion is multipolar. Of course, it's not as simple as that in biology. It's not like everything is black and white. It's either primary cement or secondary cement. There is a mixture. If you have an injury, you will have secondary cement coming into areas where there was perhaps only primary cement before to repair the tissue. So here is a beautiful microscopy example of how you can see this. Around here, this would be this A kind of stripe here. So for your orientation, again, this would be the dentine in the middle. This A kind of zone here is cement, but I don't see any dots in it. These dots are cell bodies, so this would be acellular primary cement. This whole B layer here with all these cell bodies, dots in it, is secondary cellular cement. So they are mixed together. Bear that in mind, that it's not so black and white, but usually you can stage where you are in the kind of depth of the tooth by looking at which types of cement are there. Okay, so the periodontal ligament is a really important structure which develops alongside cement development, so PDL, because this is a long name, you won't use this very often. Here is your tooth, here is the bone, and the periodontal ligament refers to these, this kind of, it looks like a gap in space, but it's actually a very flexible but very strong structure that holds the tooth where it needs to be. So it is a soft, specialized connective tissue between the cementum covering the root and the bone forming the socket wall, and it supports the teeth in their sockets, allowing them to withstand forces of mastication, so you can chew. Represented kind of cartoon-wise again, so here it would be the cement that we just talked about being formed on the outside of the tooth. So this will be your primary cement, remember, one directional primary cement with no cells in it, and this is the bone. When we talk about PDL, we're talking about these fibers which join from one side to the other side, making a kind of a link between the two tissues, between the bone and between the cement. You can see it under the microscope like this. Maybe it's a little bit less obvious in the middle, but you can see, I hope, the PDL fibers sticking out from both sides, from the cement and from the bone, and becoming kind of intertwined with each other in the middle. So how does this get formed? What is this, the basis behind this? So if we go back to this diagram and to the top, when we were starting to get cement formation, the cells which are responsible for making the PDL are these green cells here. They are differentiated from the dental follicle, which turns into both the cells making the cementoblasts to make the cement, and also the fibroblasts, these green cells, to secrete the proteins of the PDL. So these fibroblasts, these green cells, all of PDL development actually is linked to the connectivity of collagen. It's a really important thing for you to remember. These fibroblasts, green cells, produce collagen fibers, lots of them, so they secrete them. Collagen sticks to collagen, sticks to collagen, sticks to collagen. It's a very connective material. So the collagen fibers secreted by these fibroblasts here connect to what are called the fibrous fringes of the cement or the bone. So these are like little waves of proteins kind of sticking out of the bone and the cement on either side. So here and here. The collagen fibers secreted by the fibroblast connect to the fibrous fringes of the cement and bone. So you'll hear this word in the textbooks quite a lot. When they've connected and grown, when they get bigger basically, then they're called Sharpie's fibers. So there are a lot of weird terminology in PDL development. Firstly, it's fibers, collagen connecting to the fibrous fringes. When they get bigger, then they're called Sharpie's fibers. So here we are again, the same. This is bone cement. Every time is bone cement. So 1, 2, 3, 4. These are already what we would term Sharpie's fibers, because they are sticking out of the bone and cement, and the collagen has already connected to the fibrous fringes which were in the bone and cement. And then it's a matter of building up more and more collagen and linking these fibers to make them longer and longer until they eventually connect with each other, and this is what you have at the end, is the PDL. I wanted to come back to, lastly, before the ending of this video, the epithelial rests of Malacez. Epithelial rests of Malacez, you'll see them in your books, refer to these, what in this cartoon are blue circles of cells, and they are basically leftover cells from the hairs. So here would be the dentinoblasts, the dentine, cementoblasts, cement. And you can see you have these circular balls of cells, which also, if you look in an HB staining like this one here of the developing tooth, you can see these cells as well quite clearly. These are the cell rests, so the epithelial rests. Why is this important for you clinically? Is because that these rests can hang around in the developing tooth and form into cysts. So although it might seem like some very kind of obscure biological thing that you'll see under the microscope, these cells might actually be something that you run into in your clinical practice as future dentists, because they can grow and develop into cysts and have an active role in periodontal ligament repair and also regeneration. So bear them in mind in your future life as dentists. So finally, that's the end of this Kennisclip video for you. We've covered quite a lot of complex material. So root development from dentinogenesis, cementogenesis, the hairs, primary and secondary cement, the locations, functions, how that works or not, trapping cementoblasts, which one is faster, how it looks under the microscope, and then also periodontal ligament development. So I hope this has been clear enough for you. Thank you for listening, and I will see you in your TBL session very soon. Your next Kennisclip from me will be about tooth eruption, so the process of the teeth coming into the oral cavity, the gingival sulcus, which should hopefully be fairly easily for you to understand. And then there is, I believe, some references on the next slide. Information sources, again, should be more or less what you have in your textbooks, and also I quite often use this source here, Pocket Dentistry. Thank you for watching.